Hello students, Dr. E here. Welcome to the course, and this will be a quick review of Chapter 4. So first off, we'll be talking about metabolic processes, and metabolism is, if you think of all of the chemical reactions that happen in the body, if you add them all up, that is what metabolism is. So your cellular metabolism are the chemical reactions that happen inside of each individual cell. Collectively, again, together, they're known as metabolism. There are two different kinds. You're either building up something, so you're taking smaller molecules and putting them together, kind of like taking bricks and putting them together to make a bigger house. That is known as anabolic reactions or anabolism. And that needs a lot of energy. Again, going back to that building a house example, when you take bricks and you put them together to build that house, you need a lot of energy to do that. While catabolism or a catabolic reaction is where you're breaking down that bigger, larger molecule into smaller ones, and that will release energy. So again, if you think of that building that you want to demolish, it'll release a lot of dust and energy when you do that. So again, both of them together are considered metabolic reactions. Anabolic ones are the ones that need energy and they are building bigger molecules, while catabolic reactions are the one that release energy and you are taking that larger molecule and breaking it down into smaller ones. So first off, we'll talk a little bit about anabolic reactions, and as we said, those are needed to make bigger molecules. So that's what you will be doing when you're trying to build muscle or build a growth, leading to the growth of the cells. And like we said, these need energy. And as you guys all know, the energy molecule that the body knows or uses is an ATP molecule. And the example is, for example, you've got dehydration synthesis. And dehydration synthesis would be if you have these two smaller monosaccharides, they are not attached at this point, but if you remove that water molecule, that H2O molecule, now these two smaller molecules are kind of holding hands. There is a bond between the two monosaccharides, and you've made what is known as a bigger disaccharide. So saccharide pretty much means sugar. Mono means one. So this is one molecule of sugar and there's another molecule of sugar. And you remove the water molecule, that blue highlighted molecule, you've removed that. That is known as dehydration. In order to synthesize a bigger disaccharide, so di means two sugar molecules. So you've made this bigger molecule made out of these two, and that is known as dehydration synthesis. So synthesis means to make something, dehydration means to remove the water, and that's what you did in this chemical reaction. You removed the water, so you made that bigger molecule plus water is released. These are just other examples of anabolic reactions. For example, you can take this example up here to make a triglyceride. So you have a glycerol molecule, and then you've removed these three water molecules so that these three fatty acids can attach. So now you have this bigger molecule of a triglyceride, and you've released the three water molecules. Now for catabolic reactions, that is basically the reversing of that reaction that we just did. So in anabolic reactions, we did what is known as dehydration synthesis. So in order to break that bond that we made, we would have to reinsert that water molecule, and that is known as hydrolysis. So to hydrolyze something means to add that water molecule, and we're going to do that when we want to decompose or break down carbs or proteins or fat, which are lipids. So you can see here, in order to break down that bigger molecule, again, we're going to break that bond right here, insert the water molecule back. So you broke that disaccharide into two monosaccharides one more time. Here you can see other examples of catabolic reactions. So right here in the triglyceride, all you have to do is reinsert these 3 water molecules right here, and you will break down that big triglyceride molecule into 3 fatty acids and 1 glycerol. You can also see the same thing down here in the protein reactions. So in order to control all of these metabolic reactions, they don't just randomly go on and off throughout the body. They're all controlled with enzymes, and these enzymes are controlled by other factors. So there are lots of factors that kind of contribute into the control of the metabolism in the body. So there is a balance between them, and we're going to be talking about how these enzymes control and how that balance happens. So to look at an enzyme, enzymes are what are known as protein catalysts. So an enzyme is a protein molecule. It is a globular protein, meaning that it is a huge molecule of protein. It is a 4D protein, and it catalyzes different reactions. Each protein or each enzyme is specific to a reaction. So for example, the enzyme that catalyzes the reactions for glucose are specific to glucose. The enzymes that catalyze the reaction, say for triglycerides, are specific to that triglyceride molecule, and so on. There are no overlapping of enzymes. Enzymes are very specific, and they are important to lower the activation energy needed to start these reactions. Meaning, the more the enzymes you have, that when the enzyme helps these chemical reactions, that means your body can perform these reactions with the least energy needed. The enzymes are not consumed in the reaction, meaning that you can recycle them. So at the end of the reaction, the enzyme remains intact, and you can reuse that enzyme all over again. And they kind of fit like a puzzle. So how these enzymes know their substrates would be by the shape of the enzyme. So you can see down here that big molecule is your protein enzyme, and these are the 2 substrates. And substrates are the molecules that are going to enter into the reaction. And you can see here that they both fit just like a jigsaw puzzle right on what is known as the active site of the enzyme. And then at the end of the reaction, you have your product molecule, where both of these are now bound together. And the unaltered or the unchanged enzyme molecule can be reused to perform the reaction all over again. So there are factors that affect your enzyme action. For example, if you change the shape of the enzyme in any way, shape, or form, you could see that that reaction would not be able to go forwards because of the change of the active site. So if the active site isn't, doesn't fit into the substrate molecules, you've technically stopped that reaction from happening. So things that can change the shape of an enzyme, you've got excess heat, radiation, electricity, certain chemicals, changes in pHs or the extremes of pH, so being too acidic or too alkalinic, and some poisons. Denaturation means that you've inactivated the enzyme due to an irreversible change to that protein. So for example, if you take an egg and you boil it, you've now coagulated that protein, and that's exactly what happens to enzymes, that if the body temperature goes a little bit too high, so like 104, 105, where things get very dangerous, irreversible damage can happen to proteins, specifically to these enzymes, and now that they're irreversible, that means that person will not most probably survive that because of these, the body is unable to perform any of the chemical reactions. And a metabolic pathway means that these reactions occur in a certain order. It's kind of like when you're mass producing things, they have to occur in a certain order. The enzymes act in that certain order. So you got substrate 1, 2, 3, and 4, and then you've got your end product. And each enzyme, as you see, there are different enzymes for each step And each enzyme is specific for that substrate. So enzyme 1 is going to act or going to react with substrate 1. Enzyme B, its substrate would be this number 2, and so on. In order to control this whole reaction, you will, the one that is the, that will control all of this reaction would be that very first enzyme. If that enzyme isn't there, or there's a defect in that enzyme, you pretty much, limited how much of the product you can make, or maybe not even make any of the product at all. In order to name these enzymes, so really don't name them in the body enzyme A, B, C, and D, but they're named according to their substrate. So for example, if an enzyme is going to break down lactose, the enzyme would be named lactase. So any word that you see as you're reading the textbook or any other scientific journal, if you see a word that has A's in it, A's means it's an enzyme, and the substrate would be the prefix of that word. So another example would be sucrase. So sucrase, A's means it's an enzyme, sucrase means that the substrate would be sucrose. For metabolic pathways, as we said, they have to occur in a certain sequence. You cannot skip from one sequence to the next. And then your rate-limiting enzyme is always your first enzyme in the sequence. And that's how negative feedback happens. And we're going to be talking about feedback as we go. Negative feedback means that if there's nothing preventing that reaction from occurring forever, it actually will happen forever. And you're going to have tons and tons of that end product. In order for the body to limit that from happening, when the body has enough of the end product, by inhibition, it will inhibit that very first enzyme and prevent that reaction from occurring. So that is known as negative feedback mechanism, and where the very first enzyme is the rate-limiting enzyme. Now for cofactors and coenzymes. So a cofactor and a coenzyme, these are kind of co-workers to the enzyme. So the enzyme is the main worker, but sometimes it needs a little help. So the cofactor is a non-protein part. Remember, we said that the enzyme is made out of protein. A cofactor would be a non-protein part. It could be an ion, for example, maybe a sodium ion, a zinc molecule, or something of that kind of sort. That just helps the enzyme in different ways. There's also a coenzyme, which from the name is a co-worker or a helper to the enzyme. And it's different from a cofactor because cofactors are non-proteins. They're usually ions, as I said, while the coenzyme is usually an organic molecule. For instance, vitamins, those are the most common coenzymes. An organic molecule means that it has lots of carbon and lots of hydrogen. And we need these vitamins, although we need them in very, usually in smaller amounts, but we need them because they help these enzymes do their work. So how do we get all the energy for the metabolic reactions? We get them through a process known as cellular respiration. And cellular respiration doesn't literally mean that the cells are breathing, but what it means is that these cells are taking glucose and burning that glucose molecule to make ATP. And again, we use that ATP as a source of energy for everything in the body. So let's introduce what an ATP looks like. ATP stands for adenosine triphosphate. And you can see here the adenosine part is made of an adenine and a ribose sugar and 3 phosphate molecules, hence the name adenosine tri or 3 phosphates. The energy bonds are these squiggly lines between the 3 phosphates. So you see there are 2 squiggly lines, meaning that they're 2 high energy bonds. And in order for the body to release energy, it has to break these bonds, releasing that energy to use the energy in whatever activity it needs. So here you can see the the ADP molecule, or the ATP molecule up here. And whenever you need energy, again, the cell will release the energy that is found in one of these squiggly lines, and that will lead to what is known as ADP, or adenosine diphosphate, or 2-phosphates, and you'll release that phosphate out here. So you'll make the ADP plus phosphate, releasing all the energy that was in that link right here. When you want to build back that ATP, you'll take the ADP molecule, add the phosphate to it, and kind of store all that energy back, making back your ATP. And that process of taking the ADP and adding the phosphate to it to convert it back to ATP is known as phosphorylation. And this kind of happens in a cycle, for the cell to keep on using and remaking the ATP. So like I said, all of that energy is held in all of these chemical bonds, and the energy that comes out, 40% of the energy is released as chemical energy, while 60% is released as heat in order to maintain body temperature. So that 40%, that is the part that we are going to harvest and store in the ATP molecules. 60% of the heat that we make through burning of the glucose is going to be released as heat to keep our body temperatures. So now we'll talk about cellular respiration. And as I said, cellular respiration basically means burning of glucose molecules. It happens in 3 sequences, and they have to happen in this order. First step is glycolysis. Second step is citric acid cycle. Third step is the electron transport chain, or oxidative phosphorylation. And as you can see, the first step is anaerobic, means it doesn't need oxygen, while the second and third steps, they do need oxygen. The first step, or glycolysis, occurs in the cytoplasm. These two, they are aerobic, and they both happen in the mitochondria. As you would imagine, because we're not using oxygen in the first step, we're not going to make a lot of ATP. Most of the ATP is going to be made in the second and third, in particular, the third step down here. So in the next few slides, we're going to be talking about each step individually, and what happens in each step, where does it happen, how much ATP are we making, and so on. Okay, so cellular respiration happens in a cycle. So we talked about a metabolic pathway that was linear in a line, but cellular respiration happens in a cycle where you start off with substrates that enter the reaction, and it goes in this cycle, and your end product is going to is also the substrate that you need in order to start the whole process all over again. The best example for this cycle or cyclic kind or cycle kind of reaction is your citric acid cycle. As we said, cellular respiration there are 3 different steps. Glycolysis happens out here in the cytoplasm. So if you imagine this as a huge cell, that is your cytoplasm out here, and this represents a very huge mitochondria. So your first step, glycolysis, happens in the cytoplasm, and you see it only makes 2 molecules of ATP. One of the products, though, is 2 pyruvic acid molecules, and these pyruvic acid molecules enter the mitochondria, and they change into acetyl-CoA. Acetyl-CoA is then going to bind with oxaloacetic acid, and both of them together bind together to make this citric acid, and that will start your citric acid cycle. As you see here, the cycle, all of it occurs in the mitochondria. We are also only making 2 ATP molecules, so you see there's not many molecules being made. But then during glycolysis and the citric acid cycle, we are harvesting you can see here high energy electrons being harvested or collected, and all of these high energy electrons are going to enter the very last stage of cellular respiration, also known as the electron transport chain. So we're going to take all of these electrons and they will all go through the electron transport chain to make at the end between 26 to 28 molecules. So you see again that most of the ATP is being made in that third stage of events. So for cellular respiration, again, we're going to burn the glucose. So you always need a supply of glucose and you need a supply of oxygen. Your products are going to be carbon dioxide. You're also making water. You're making ATP. So remember, only 40% of the energy is going to be stored as ATP, while 60% of the energy you make by burning the glucose is going to be released as heat. The two different reactions that we have, we got the aerobic reactions, and those are steps 2 and 3. Those need oxygen, and those make most of your ATP. The anaerobic reaction, that's the first step, that's glycolysis. It does not need oxygen and makes very little ATP, 2 molecules to be specific. So first step, I'm actually going to go to the figure here. So your first step, as we said, is glycolysis. You see here the glucose molecule, and all of this again happens in the cytoplasm. The first phase of glycolysis is phosphorylation. Phosphorylation means to add phosphor or phosphate molecules. So you see here glycolysis, we're going to take the 2 ATP, we're going to take phosphates from ATP molecules and add, there's one phosphate and there's another phosphate. And then your second stage is splitting or cleavage. So we're going to split this big, what is known as 6 carbon molecules, and split it into 2 smaller molecules. Each molecule has 1, 2, 3 carbon atoms. So these dark gray circles represent carbon atoms. So again, your splitting phase is where we're splitting that big molecule into 2 smaller molecules. And then the third stage would be the ATP formation and release of electrons. So as we said, we have to harvest these electrons and take them to stage number 3. The carriers for these electrons cannot go on their own. They have to be carried by carriers. We have 2 different kinds of carriers, NAD and FAD. For glycolysis, the carrier here is NAD, as you can see. So this NAD is going to change into NADH. And your final result is making of that pyruvic acid. Now that pyruvic acid has 1 out of 2 ways to go. If there is enough oxygen, the pyruvic acid enters into the mitochondria and starts the citric acid cycle. If there's not enough oxygen left, you can see their oxygen is crossed over, so not enough oxygen is found in the body. That pyruvic acid is going to burn even further and make lactic acid. And lactic acid is what is responsible for any muscle acheness, and if you exercise too hard and you wake up the next morning with muscle soreness, it's the lactic acid that's causing that soreness. So your anaerobic reaction, like I said, in the presence of oxygen, the NADH and H will deliver the electrons to the transport chain. In the absence of oxygen, there is no electron acceptor. The NADH, I actually prefer using the picture, really. So to go over all 3 steps all over again, that glucose molecule is going to be phosphorylated. That's your first step. So we've added the 2 phosphates. Second step is the splitting or cleavage. So we're going to split this bigger molecule made out of 6 carbons into 2 smaller ones. Each one is made out of 3 carbons. Your third stage, that would be your ATP formation. So you're making 2 molecules of ATP. And you are going to also make 2 molecules of pyruvic acid. And we have to harvest, we're going to be harvesting some of the electrons, And the carrier to harvest these electrons are NAD. You see the NAD is changed into NADH plus H, and same here as NADH. These NADHs will go to the mitochondria into the electron transport chain, which is the third step, while the pyruvic acid has one out of two different options, depending on the presence or absence of oxygen. So if there is enough oxygen, pyruvic acid will enter the mitochondria and enter the second stage of events, which are the citric acid cycles. If there isn't enough oxygen, that pyruvic acid will change into lactic acid and cause soreness. We're going to learn how the body gets rid of that lactic acid, because it is technically considered not a poisonous poison, but it is considered something that the body has, a waste product that the body has to get rid of. So you guys can go ahead and read these. This is everything that was on the slide that we talked about. If you have any questions about them, just feel free to email me anytime. Your second stage is the citric acid cycle. So now the pyruvic acid that we've made from glycolysis, again, if there's enough oxygen, will enter the mitochondria and will change into acetyl-CoA. That acetyl-CoA combines with another molecule known as the oxaloacetic acid. Oxaloacetic acid is already present in the mitochondria, and both of them combine to make citric acid. Now that citric acid will enter a cycle of several chemical reactions, and hence the name citric acid cycle. Okay, we don't really have to know all of these intermediate chemicals, their names, but you do have to be familiar with where the pyruvic acid came from. What are the conditions for it to enter the mitochondria? Is it presence or absence of oxygen? And it changes to acetyl-CoA and combines with oxaloacetic acid to make citric acid, and that's how the citric acid cycle starts. At the end of this cycle, you're making ATP. You have also harvested electrons and hydrogen ions. Okay, and your carriers are here, NAD and FAD, and you've produced carbon dioxide. So this is where carbon dioxide is produced by the cells, and that carbon dioxide has to go through the blood, go to the lungs, so that we can exhale that carbon dioxide. Now for the very last stage, or the electron transport chain, again, this happens inside of the mitochondria. It happens in the mitochondrial walls, to be very specific. And you can see all of these electrons. These walls were kind of shelves. You can go back to the description of what a mitochondria looked like. There were shelves in the mitochondrial walls, and all of these high energy electrons are going to kind of whirl around and release lots of energy. Now the body harvests all of that energy, and to change all of the ADP, phosphorylates them and makes ATP molecules. At the very end, that glucose is fully burned. All you have left are hydrogen molecules, and you can't leave them there because that would lead to acidity of the blood. So these hydrogen molecules are going to combine with oxygen, and your final molecule is a water molecule. So that you've started off with glucose, you've burned it through those 3 different stages, and you end up with a water molecule at the very end. And we call here that oxygen, it is called the final electron carrier. So remember the carriers that we talked about, NAD and FAD. At the very end, your final electron is going to be carried by oxygen to make a water molecule. So right here, this is an overview of your cellular respiration. I'm going to let you guys go over that on your own. Now for carb storage. Carbohydrates are stored in the body as glycogen or fat. So when we eat up carbs, okay, they are hydrolyzed, so they are digested into monosaccharides, and that's how we can absorb them. So we absorb these monosaccharides, and there are several pathways for the monosaccharides to go through. They can go through catabolic pathways, so we'll burn these monosaccharides, we'll burn that glucose to make energy, carbon dioxide, and water. or the monosaccharides can go through anabolic pathways. We can store glucose as glycogen, okay, or triglycerides, which is basically fat. So as you all know, if you eat too much carbs, that will eventually translate into increase in the size of the fat cells. Or another anabolic pathway that it can go to would be to make amino acids and build up protein. So these are your three ways for the monosaccharides to go through. One of them is catabolic for energy. Two would be anabolic. Either we're storing it as glycogen or triglycerides, which is fat, or we are using it to build up amino acids and protein. This here shows you a summary of the catabolism of proteins and carbs and fat. So all of these reactions cannot just happen on their own. There has to be a manual that explains all of this, that has all of the steps. And that manual is known as the genetic material, and that genetic material is found in the DNA, or deoxyribonucleic acid. So the DNA stores the instructions on how to make enzymes, how to make blood proteins, how to make any proteins, whether it's muscle, connective tissue, epithelial tissue, antibodies. If you can think of anything that's in the body, it needs a manual, and that manual is going to be found in the DNA. Now, just some parts, different concepts that we need to understand in order to understand what we're talking about in the next few slides. So first off is genetic information. That is everything that is found in the DNA. So that is the whole manual that makes your DNA sequence. A gene is just a portion of that DNA. So a sequence of DNA that has information to make just one protein. That gene, however, has parts that code for a protein and other parts that do not code for a protein. So the part that codes for a protein is known as the exome. The other parts that do not code for a protein are there to control how the protein is made, how much of that protein is made. So all of that together would be your genome. Would be the gene. Okay, so again, the gene is made out of parts that are going to be made into proteins, and that is an exome, and other parts that we'll talk about later, actually in this chapter, that do not code for a protein, but if you put them both together, it'll make a gene. A genome is the complete set of genetic information that is found in a cell. And gene expression means which genes are we going to express in which cells. So I'll give you an example of that. First off, know that all of the genetic information is found in each and every single cell of the body, which means your liver cell has information on how to make nails, but our liver does not make nails, obviously, or hair. That's because these genes are not expressed in the liver. So gene expression means that although the gene is there, it doesn't have to be expressed. But the genes that make nails or hair are going to be expressed where they should be. So in the fingertips, all over the skin, and so on. So the presence of the gene does not necessarily mean that it has to be expressed. If you have any questions about this, again, please let me know. I'll explain this a little bit further. So structure of DNA are made out of a chain of nucleotides. So your building block is this nucleotide right here, and it has a sugar base, and that sugar is deoxyribose. It has a phosphate, and it has a nitrogenous base. So it's got a sugar, which in the case of DNA is a deoxyribose, and a phosphate, and a nitrogenous base. The nitrogenous base in DNA, you're going to choose one out of these four, so adenine, cytosine, guanine, or thymine. And it kind of resembles a ladder. So I'm going to show you this picture right here. These are your nucleotides, and these are other nucleotides. And you can see here that these bases are connected by very weak hydrogen bonds, and that's why the bonds are found in interrupted or dots, because they're very weak bonds. As soon as you heat them up, you can break these bonds very easily. These bonds are going to have to happen between two complementary bases. So an adenine will always bind to a thymine, and a cytosine is always going to bind to a guanine, and that would be throughout the whole sequence of DNA, and that's how that's is really the genetic code, would be reading the sequence of bases on these nucleotides. You can also see that it is double-stranded, so this is one strand and there's another strand. If you compare that to RNA, which is ribonucleic acid, you can see here that it is a single-stranded molecule. The bases are a little bit different. So we have adenine, guanine, and cytosine. Those are the same, but there is no thymine in RNA. It is replaced with uracil. And the sugar is obviously different. So in DNA, it's a deoxyribose. In RNA, it is a ribose sugar. So here you can see the structure again of DNA and how it is twisted into what is known as a double helix. We've already talked about these complementary base pairings where the adenine and thymine are always bind together, and cytosine and guanine always bind together as well. You can see that they're very tightly wrapped around these little histone proteins, so that at the end they look like these X-shaped chromosomes. Now, if you could go to the DNA replication file that is posted for you on Canvas, we'll talk first about DNA replication. DNA replication means that you're going to make a copy of the DNA, and you need to make copies of DNA when the cell is about to divide and make another copy of itself. So your steps in your DNA replication would be, first off, you'd have to break these bonds. You have to break these hydrogen bonds that are found here. You have to unwind this, and then you have to read these bases and bring in the complementary bases again. So you'll end up with two copies. You start off with this one copy, but you'll end up with two copies. So again, your steps, first off, you have to break these hydrogen bonds. You'll untwist or unwind, as you can see it here, and then you are going to read these nitrogenous bases and bring in the complementary bases. At the very end, the enzyme that you are going to be used is known as a DNA polymerase enzyme in order to make that huge DNA molecule. In order to make this genetic code, the genetic information, now to our next topic, and that would be protein synthesis. Protein synthesis means we're going to use the genes that we're reading off of the DNA, read that genetic code, and then make a protein out of it. So the genetic code, as we said, are those nitrogenous bases found on the DNA molecule, and we are going to read them in threes. They are read in what is known as triplet codes. So a sequence of 3 nucleotides, those represent an amino acid, and the body reads these in codes and translates that or uses that in order to make its protein. The only problem is that the DNA is found in the nucleus, and if you guys remember, protein synthesis happens in the ribosomes, which is out in the cytoplasm. The DNA cannot leave the nucleus, so it has to send out a messenger that carries that genetic code, and that messenger is known as the messenger RNA. You can see here an RNA. We already talked about an RNA. We said it is a single-stranded, made out of a single strand of nucleotides. The sugar is ribose sugar. The phosphates are the same, and you have these nitrogenous bases, but remember we have adenine, guanine, and cytosine. but instead of thymine, we've got uracil, represented by this U right here. For the complementary bases, the C and G are complementary while the A and U are complementary. So your very first stage again, or first step in making a protein would be transcription, which means the process of making a copy of DNA, changing it into a messenger RNA, and this process happens in the nucleus. You'll take that code, the messenger RNA has the ability to leave the nucleus, and that process happens through an enzyme known as RNA polymerase enzyme. So in order to talk about these steps of making a protein, I'm actually going to go to this figure right here, and this yellow part represents the cytoplasm while the nucleus is represented by that blue. Here you can see the DNA, the double helix DNA. The first step, as we said, would be transcription. So we are making a messenger RNA. Remember, we talked about how the DNA cannot leave the nucleus. We have to make a messenger that we called a messenger RNA. And that process of making a messenger RNA from your DNA is known as transcription. So as you see, it happens in the nucleus, and then that messenger RNA will leave the nucleus through the nuclear pore. Now that messenger RNA will attach to a ribosome, and a second stage, or the second step of protein synthesis happens, and that is the step of translation. So now that ribosome is going to translate the messenger RNA into a protein. And again, it reads it in threes, as you can see here. So that ribosome is known as the ribosomal RNA. It'll read the messenger RNA in triplets, in threes. That is known as your codon. And then this transfer RNA, this funny looking molecule here that is known as a transfer RNA, has what is an anticodon that will be the complementary of these codons. So for example, if the codon was on the messenger RNA, if it was AUG, the transfer codon would have an the transfer RNA would have an anticodon of UAC. Okay, and then that would code for a certain amino acid, and the transfer RNA will look for that amino acid in the cytoplasm, bring it in, and then it'll go on to read the second codon, and so on. As you can see here, the process of making the protein keeps on going, the translation process. So again, we're translating the code into a protein, and you can see here that each one of these represents an amino acid, and then there are peptide bonds in between. So the transfer RNA will bring in the amino acid, attach it to the forming protein, and then go off its way to find other amino acids and so on. There is a video that I posted for you guys on Canvas. I strongly recommend that you watch it. It is a video that represents the process of making a protein, and the discussion board for this week really builds on it. So that really talks about the process of protein synthesis, how the only way the body can know where to start reading the transfer RNA is by what is known as a start or an initiation codon, and that would be AUG. That codes for methionine. So if we go back to this picture, the ribosome will read the messenger RNA, and as soon as it finds that the start codon or the AUG, it knows that this is where I need to start making the protein. And it'll keep on going down until it reaches what are known as stop codons. Okay, there are 3 different stop codons, and again, that's how your ribosome knows that this is where I'm going to stop, and I'm done making this one protein. There's no reason for anybody really to know this table. You just want to make sure that you're familiar with the start or the initiation codon, and that is AUG. and the stop codons, and those are UAA, UAG, and UGA. Again, here you can see the ribosome reading the messenger RNA, and here are these little transfer RNAs bringing the amino acids. They bind through the peptide bonds, and then the transfer RNA leaves and goes to find another amino acid, and so on. And you can see here the protein chain is being built, and it's getting longer and longer as it keeps on going until again it reaches a stop codon and it stops. So this is really just basically talking about the same things. I recommend that you go ahead and read these on your own and let me know if there are any questions. This is a table that shows all of the steps of protein synthesis. So step number 1 is transcription, as we said, that happens in the nucleus. And that is the process of making the messenger RNA. The messenger RNA will leave the nucleus and go to the second step of translation. That happens in the cytoplasm. And here you've got an interaction of the messenger RNA, ribosomal RNA, and transfer RNA. Know that the codon, make sure you're familiar with where the codon is, where the anticodon is. It helps to know that the codon is on the messenger RNA. The anticodon with a T is on the transfer RNA with a T. Be familiar with your stop and your start codons and so on. Again, don't have to be familiar with how did this this table of translation make. And an easy way to remember step 1 and step 2, transcription is alphabetically, if you put them in alphabetical order, transcription would be first, followed by translation, and that's the same order of events that happen inside of the cell. This concludes a summary of chapter 1. Again, feel free to email me with any questions. Good luck.